professor. And so good to be back here and to see so many familiar, friendly faces. Folks I haven't seen. Back then I was talking about permeability calculations. We sort of stopped doing those a while back. So today is going to be a bit different topic. There you go. Now I can hear myself. I'm going to tell you all about our efforts to use molecular simulation as a multi-scale computational microscope for drug and immunogen design, and also for some other fun things. Okay, so I think in this audience, I really don't have to belabor the point that simulation, and now together with AI, is really a new form of experiment. I think that's the right way for us to look at these types of calculations, that we are partners with experimentalists, and can hold our own in many cases. I think that's important to remember. So, you know, nowadays what we can do is we have all this amazing data sets taken from so many different instruments across a range of, you know, fields. Could be mass spec, could be structural, biological structural instruments, could be any number of things, genomics, glycomics, fluorescence microscopy, and we can actually use computing and these types of physics-based models to combine many different types of data sets together into one cohesive model that we then can approximate down to its many atoms, like all of you are, I'm sure, very familiar with, and then use physics-based simulations. Some people will even refer to these in some instances as digital twins, and also together with AI. Here, this is just our sort of basic equations that we use. And actually, it's ironic. So normally, I work really hard to take the equations out of my talk. I definitely noticed this morning, well, it's a methods conference, so there's a range of, you know, more people, I think, including them, but you'll have to forgive me for removing most of them. But in any case, we can then, you know, we approximate the system down to its many atoms. We integrate Newton's equation of motion over time using various force fields and this potential function. And then at the end of the day, we do a lot of analysis, and we can actually make discoveries that impact real world things, you know, in the wet lab and our understanding of chemistry and biology. I think it's ever an exciting time to think of this as a computational microscope. I think that's how we use it and many others. And that is really powerful in terms of data integration and synthesis to drive new discoveries. Okay, so this is a multi-scale microscope. As all of you know, we can cover quantum behavior at the smallest time and length scales, and then we can go to nanoscale, mesoscale, and macroscale. I'll touch on all of these different scales in the talk today. The first realm that I will just take a little, like, dip the toe into is in quantum. Mostly what I'll focus on are sort of the larger scale system work that we focus on. So you already heard about this. So when I got invited to speak here, I got really excited because most of the time I get invited to talk about the big simulations, which I love. But we also have had for a number of years, almost since the beginning of my lab, well, since 2013 or so, we've been developing a program called Seeker, Simulation-Enabled Estimation of Kinetic Rates. And you heard Anupam Anand Oha, who's now a fellow at the Flatiron Institute. He presented this morning the work in detail. I was excited because I thought, oh, for energy methods, they're asking me to talk about Seeker. But then actually when I clarified with the organizers, they were like, yeah, that's great, but we'd really also like to hear about your big simulations. So, but I had to put a couple of things in, just, and I know you already heard from Anand, but I just, this is cool because it links to this whole idea of where we want to go, where we are going with molecular simulation in conjunction with, you know, not just using molecular dynamics, but also Brownian dynamics at, you know, longer time and length scales, and then moving to reaction diffusion type models and continuum methods and so forth. So we won't cover all of that today. We're going to stay in molecular mostly and Brownian dynamics. But so this program that we've developed actually is multi-scale in that at just when we're, so the focus is trying to, or the point originally was to try to really develop a method that could quantify drug binding kinetics and unbinding kinetics from receptors. And so our idea was to use rigid body Brownian dynamics when the two molecules are far apart. And when they get closer together, then engage fully flexible molecular dynamics simulations. and to use milestoning as a theory to bridge these regimes. You heard also now we've integrated, as I'll talk about, QM, so some quantum mechanical corrections in the bound state to help us get around some of the limitations of the fixed charge force fields. And the cool thing about this, which we like dynamics, we like kinetics. This gives us an estimate of kinetics, and it also lets us get K on, K off, and delta G of binding with one set of calculations. Okay, and so over the years, we started with toy systems like host guest. And over the years, we've been able to, if we look at receptor complexity and ligand complexity, we've been able to go from these like small model systems to systems that actually people might be interested in real life. And so there's a whole bunch of papers. Anand did, I think, a really nice job going through it. I just want to say, cover the last few years. Maybe this is the better microphone for me. In a collaboration with Johnson & Johnson, we set out a few years, published 2 years ago. It actually took us some time to get this out, but we were able to use this method to look at JAK2 and JAK3 inhibitors, not only getting some fairly decent estimates for residence times, here's theoretical versus experimental, but also we were able to mechanistically understand selectivity between the two for some of the small molecules. And then one of the things, as we tried to expand this approach, we went to larger data sets like the HSP90 data set with all of its many molecules. There's many more reported with residence time data than we're showing here. But one of the things that we were interested in, the ones that we were getting wrong, and there's of course a lot of reasons why one gets things wrong in this field. Many of these points have already been elaborated. But with HSP90, one of the cool things that Anand did was actually include quantum mechanical correction in the innermost milestone, again, to get around the limitations of the fixed charge force field. And we saw that for some of the ligands where the force field was an issue, because it doesn't solve all the issues that we have, it actually brought those into line with the ones that were working. So we were happy about that. That was published also in 2023. And then, one of the other things that's a challenge is slow receptor dynamics. So then more recently still, we integrated metadynamics together with this as a way to look at the slower receptor motions. And we explored this in a collaboration with Shionogi and tyrosine 3-amine kinase, and we were able to improve our estimates of the residence times and also delta G of binding. And then this work he mentioned also. So one of the, usability is obviously an issue, and we're an academic group, not making, software necessarily. I mean, we do make software, but it's not necessarily super easy to use. But he's recently developed an automated pipeline or workflow that will help people use this more easily, and also incorporated machine learned force fields, the one by Tradera and colleagues, Spaloma, and shown also that actually does a pretty good job of replicating what we had done already with the QM corrections. So this is not yet published. It's in preparation. It's also, I should say, in collaboration with Sonia Hansen, who's at Flatiron too. So this becomes ever more useful. Now, to date, all those targets that I showed you, they're soluble targets. And the reason for that actually, well, there's data there, but also because our Brownian dynamics package that we had been using is something called Brown dye, and it had particular boundary conditions that prohibited us to use it for receptors, for example, that are bound in the membrane. However, recently that has changed, and we've gotten now, we've hooked up something called Simulation of Diffusion and Association, SDA. This is an old Brownian dynamics program, still developed, but created in the 90s, mostly in the group of Rebecca Wade at Heidelberg, and we've now hooked that in as the BD engine, so we can start to use this now as a method to explore systems in much more complex scenes. And that's really sort of one of the core interests of my group is in developing these sort of highly detailed, complex models of biological systems that integrate structure at different resolutions, so cryo-electron tomography together with single particle and X-ray, to build molecular level scenes, for example, in neurons. So this is an axon here, and you can see that, it's really a lot of the interesting receptors in the brain and other places are in fact in membranes. So we are on the edge of being able to do that now. And I have a typo there. Okay. But now the next thing is, so that was a little bit about quantum, was the little fix that, or the nice fix that Anand brought in. Now I'm going to talk about our work at the nanoscale. And what I'll talk about, because who knows how much longer I'll be talking about COVID, because I think all the funding is really going away now, in the United States at least. But everyone is familiar with this virus. It looks like a golf ball with spikes sticking out of it. It has a membrane. It's a membrane enveloped virus. It has structural proteins embedded in the membrane that help retain its structure and resiliency as it infects cells. And then it has a so-called spike protein, which everyone is probably familiar with. The spike protein is a very important part of it because it sits on the outside of the virus. So it's the first point of contact that the virus has with human cells. So it plays a really key role in infection. And it's also a highly immunogenic molecule. So, it's important for the vaccines and so forth. And so in terms of infection, this little bit here, this is to scale, I guess, so this is the ACE2 receptor, angiotensin converting enzyme 2, which is the native receptor for the spike protein in a very empty, uncrowded membrane. So this is obviously just for visualization. This is really the handshake that needs to happen in order for the virus to infect human cells. And to me, this is really just amazing. It's just this tiny, oops, I hit the thing. Which one was it that I hit? That one, okay. It's just this tiny interaction between these two proteins that, can really cause so much damage. So I find that fascinating. So what we did and what we like to do in my group is to build these models where we combine experimental data. For example, here we have the head, or the so-called ectodomain of the spike protein. That's been resolved by many groups now with cryo-electron microscopy. But then we also have data that they can't see in these structural experiments, like glycans, end-linked glycans, which play a very important role. I'll tell you about those in the behavior of this protein, but otherwise are unseen by experimental techniques. However, we have this data from mass spec. So we can create these hybrid computational and experimental models. And and then we simulate them using all-atom MD. I'm hiding the water molecules. So that's also, you're in for it. All the other talks this morning were really proudly showing the water molecules. And of course, I have to confess, is the first things that we usually strip away when we're trying to ascertain these large-scale motions that we tend to be interested in. But of course, so we build these models as an example of the spike protein, and then we look at the dynamics, and we can learn things about the structure and function of the system. So our early work in SARS-2 showed what the spike really looked like. So on the left is what the experimental structural biologists see. They basically can see the protein. They can't see those N and O-linked glycans because they're just wiggling around too much. They're too dynamic. On the right is what that protein would actually look like if they could see the glycans. And you can see how it's a very different surface, an object that's actually presented to the body than this. And so each of these tufts, like each of these little blue tufts, represents one N-linked glycan. It's a composite image of all the space sort of that it's sampled over 1 microsecond of dynamics. So you can see that there's an N-linked glycan here and here and here and here and here. And then over time, they're moving at very quick time scales. It basically sweeps out an occluded volume of space that shields this molecule from the human immune system. And this shielding was, of course, actually well known to be the case in viral glycoproteins and other viral proteins. They hijack the human glycan-adding enzymes literally to mask themselves and go undetected in the body. But what we did not know, but what simulation allowed us to discover, was a new role for glycans in biology. And this was that when we were spinning this structure around, stuck in our kitchens during COVID, looking at the dynamics, we realized that when we rebuilt the glycan side chains please play again. When we rebuilt the glycan side chains for two of these glycans, they actually, so this receptor binding domain actually moves. So it actually hides itself and then it goes in the closed conformation, and then it also goes into an up conformation where it's revealed. And when we rebuilt those side chains, what we saw was that those glycans actually filled the void space that was created when that RBD went into the up conformation. And so what we hypothesized, showed in experiment, and then had experimental collaborators also show in experiment, was that if you remove these two glycans, you reduce the ability of the spike to infect cells and to interact with ACE2 because that domain becomes unstable. So this actually quite literally rewrote the textbook of glycobiology. They put this on the cover. This is a Bible of glycobiology. And I see this, and I really think it's a triumph of simulation because, you know, in spite of the limitations of the force fields, which we know exist, especially for small molecules, but somehow we're able to really drive, you know, forward a new discovery, opening sort of this new paradigm. That was pretty cool for all of us. So all we were doing there was just like conventional vanilla dynamics around two structurally resolved states because they had the up state from cryo-EM and they had this closed state. So we then applied weighted ensemble because we wanted weighted ensemble MD because what we wanted to do was actually look at the opening of the spike protein en route to infection. And so here you can see the RBD sort of coming open. This is the delta spike. With all of its glycans. And this was a collaboration with Lillian Chong at the University of Pittsburgh. She's a developer of WESPA, along with Dan Zuckerman. And we did this for different, for the original strain, which was circulating, and also the Delta strain. And we've done it for one or two other strains. And what you can really see is that the virus is evolving different ways of opening. It opens easier, it opens mechanically a little bit different, based on the variation that it's undergoing in sequence space. What we also discovered was that there was another glycan shown here in hot pink, which if you just looked at those original conventional MD simulations, if you look at the upstate 10 microseconds of dynamics, this glycan does not look like it's doing anything interesting. It's sitting at the side of the protein. It looks like it's shielding. That's in the upstate. If you look at it in the downstate, it's the same thing. It's sitting there on the side, doesn't look like it's doing anything interesting. But when you do the work to look at the mechanism of opening, you see a totally different story. This pink guy is actually an active participant in opening that receptor binding domain. It's actually doing sort of like a hand jive motion, if you will, with other residues all along this receptor binding domain, and ultimately sort of kicks it out like a crowbar. We went back to our collaborator, Jason, and had him and his team do some more experiments. And this glycan is even more important than the other two. And in fact, if you get rid of it, the virus can't infect cells. Okay, so then more recently, we became involved in trying to do immunogen design. And so for this, you know, here's a spike protein, you've seen it. What I told you about mostly were dynamics in what's called the S1 subunit at the top. The S2 subunit is what is inside this protein underneath the S1. And so this protein, as well as all of the other class 1 glycoprotein fusion proteins, are basically spring-loaded molecular machines that are designed to recognize a receptor on a host cell. And then the tops come off, and then these central helices polymerize upwards, and it brings the two membranes together, and then we're all screwed. So, basically, what we did here, people have been developing vaccines which utilize the whole construct, including S1, but we know that those vaccines have lost effectiveness because of this enormous amount of mutations that occur, right? So then the antibodies don't recognize it. S2, however, gives us a different story, and it has long been something that folks have wanted to develop potentially as a pan-coronavirus vaccine. It's very highly conserved across the beta coronavirus family, much more so than S1. And it is known to elicit antibodies that give broadly neutralizing protection. So it has really good immunogenicity. And there's actually several different epitopes, which I'm showing here in orange. There's one at the apex, there's some down here, there's some down in the stem, which confer additional protection beyond the ones that we heard all about with the mRNA vaccines that are on top of the S1 subunit and in the N-terminal domain. So one of the challenges has been, though, is that this S2 domain, by design, by nature's design, is highly unstable because, like I said, it's a spring-loaded machine that needs to just kind of quickly unravel to bring the two membranes close together. So, so that sort of challenged its use in a, oops, sorry, I went the wrong way. That sort of challenged experimental efforts around trying to make that a good immunogen. And in fact, so you may or may not know, but there have been a lot of people trying to design more stable constructs of the spike protein. This is to gain experimental efficiencies as well as to potentially work with the S2 domain on its own. And even in a hexapro form, what you see, it's a highly unstable molecule that, for example, they were not able to get high resolution cryo-EM structures in even the stabilized state. So sorry, I'm thirsty. So I think I got dehydrated on that plane ride. So we wanted to see if we could use simulation to come up with some stabilization and design ideas. And so what we did was we did the hardest types of simulations you can do. Actually, I kind of think they are. But we really kind of, well, I have to say this. Okay. Weighted ensemble simulations. You know, my group is a group that tries a lot of different methods. We did a lot with Markov state models. We've done a lot of different things. And, you know, we enjoy that. And we enjoy applying these new techniques that people develop to real world systems. When we learned about weighted ensemble, I have to say I was a little like, yeah, this is going to be useful. And then, you know, it led us to all these discoveries about the mechanism. And I think if you really want to know about mechanism, to me, that's where we go. We almost go straight to weighted ensemble because it's just so terrific in how it can elucidate these sort of intricate details in a way that I think other methods really cannot. But so we decided to look at the opening mechanism. We tried at first with conventional MD, of course, who wouldn't? But it was too slow. I mean, it was unstable, but still too slow for conventional MD. So we used weighted ensemble to open it. And our thinking was, if we could understand how it opened, we could then potentially design in contacts that would stop the opening or slow it down. So what you see is it's sort of, here's going slow-mo, it's opening. We do get it to open sort of down, as you'll see, down some rungs of the ladder. Here's a picture of it sort of colored through, I call these sort of like hockey stick plots, but basically we have our collective variables. A lot of folks working on efficient ways to predict collective variables. We do it the old-fashioned way, which is to try a bunch of different things based on our intuition and knowledge of the system. But there's, you know, I think obviously a lot of opportunity there. So here you can kind of see the space that we're walking through or going through as it opens. And what we found was that it opens asymmetrically, sort of from top down in sort of a zipper-like opening manner. And when you start analyzing these trajectories, so this is S2 that I just showed you opening. I'm going to focus in these three purple, what we call the central helices. These are the core of this S2 spike. They're the core of many of these glycofusion proteins. And when you look at it, you can almost see that the residues almost form like rungs on a ladder, right? So here's the bottom rung, the second rung, et cetera, and the top rung. And so what we found was that it was opening top down, as I mentioned, sort of in a zipper-like. And when here's on the right is basically a contact plot where if you see yellow, it means there's basically no interactions. If you see red, it means there's interactions. Okay, and so immediately what you see is that in sort of the wild type construct, or we actually had started anyway already with Hexapro, but our original construct that we were shooting at to improve with the experimentalists, the top basically is just designed to not, it doesn't have any contact. So this jumped out as a spot where, and also this rung jumped out as a spot where we might be able to introduce something that would introduce contacts that would stabilize the closed form, as well as down here, sort of the third rung. So there was in these two positions. And then what we decided to do, we tried a few different things. This included free energy calculations, which I'm actually not showing you here just because I normally don't talk about them. And I did not update this slide, but I also have too much that I want to tell you. But we ended up substituting tryptophans. a ring of tryptophans. You can see them here. And when we did that, what we found was so we did it, we made the prediction based on the unraveling, the opening, and also the free energy predictions. And then when we put them in, we actually put them in silico and rerun dynamics again. And we could see that it was slower to open and much more stable. And in fact, it's not just that it makes contacts in this one ring and that first rung of the ladder, but it actually reorganizes a network also, not laterally, but vertically. So it really sort of strengthens a big interaction network. And then the experimentalists just made these substitutions, and what they found was they worked on the first shot, which is kind of amazing. We've made, because of that, the most thermostable construct of the spike proteins, and we have carried the work on for immunogen design. Unfortunately, it does stabilize the protein. They're able to get the high resolution structure, but the apex epitopes that we were targeting with this stabilization immunogenically were not winners. I mean, they did a little bit, but not enough to actually go forward into like the next vaccine. So this probably got shelved, but was a nice example of using weighted ensemble methods to do immunogen design. Okay, now going into the mesoscale. So I've long been in love with these big systems and trying to understand not just a protein alone, but how does a protein move and its function in these much more complex scenes. And so some years ago, this is actually now influenza virus. We had built these larger scale simulations. So the simulations of the spike protein that I'm showing you, they have about 2 million atoms. These simulations of the virus, of the flu virus, have about 160 million atoms. So they're a bit larger. And what we do is we take the cryo-EM maps, and then we take the high resolution bits, and we put these high resolution hemagglutinins, and so hemagglutinins are in blue, the neuraminases are in red, and the glycans, this one's not as glycosylated for very interesting reasons, but we show them here in yellow. And then we ran all we created 2 different strains, because you can do that. We ran this using NAMB2 on Blue Waters, which is a very old machine now. And then Oak Ridge, also old. These machines have gone away. But we got about half a microsecond of sampling for these large systems. If you're thinking, that's not very much, Rommie, what are you going to learn? I don't know. So we opened it up. We started looking at the data. What we saw, so this is the dynamics in this complex scene. I'm bringing your attention to one of the 273 hemagglutinins on the surface, just as an example, because we have to start somewhere. If you look at this guy, you might start to see this domain kind of splaying open. It's like it went open and then it came back. Let me show you another view of that. Now we've isolated one of these molecules. This is the hemagglutinins. So this is basically the spike protein, but for influenza. And from the side, you can see it kind of opening, and here's the top view. It's definitely opening. It's coming apart. When we saw, the first time we saw this, I thought, oh my God, we just wasted 8 years of our life on, that it was an artifact. But, we kept on with the analysis, and then while we were doing it, here's another just bit. So you can see sort of the time series here. So it starts, so all of the molecules that we put into the system, and I don't have time to tell you, but let me tell you now, when we build these large scale systems that have many different pieces, we start out by simulating each of the molecular pieces and making sure they are good models that can stand up to experiment on their own, and we then bring it into the bigger picture. We don't just build the whole thing like from the crystal structure state of all the copies. So but I will say all of the hemagglutinins at time t = 0 were basically closed, just like you see in the cryo-EM data, even though they had been simulated already for about a microsecond. And then we sort of do structural clustering and then and then sort of fill in the 273. They have some small structural differences as taken from the underlying simulations that we do in the individual molecules. Hopefully that made sense. So first it's closed, and then over here it starts to splay open, and it even goes super open. And so We started to worry, and we had this data since basically like 2019 or so, right before the pandemic. And then in 2020, this paper comes out, actually from some friends across the street. And what they see is amazing. They said, so this paper, what they had found was that they found an antibody circulating in a person who had been infected with the flu, so from convalescent sera. They found an antibody they called flu A20. And if you looked at where this thing would bind, you see that it's not binding on any of the outside part of the hemagglutinin. It's actually binding on what they call a cryptic epitope in the inside. And here you can see low res cryo or negative stain images here of like where it would bind. But what they write here, I'm just going to read this. It says, therefore, the HA molecules on the cellular or viral surface generally have been considered to be stable trimers, with the trimer interface regarded as inaccessible and thus not targetable by the immune response or therapeutics. I want to pause there because that's exactly right. We are biased always by what we can see. And so the cryo-EM people make these beautiful structures. Every single one of them gets into science, high resolution. You can't believe it. It's just amazing. And it's one picture of this darn thing. So we all go around thinking this is what this molecule looks like, all nice and tidy on the surface, all 273. But actually that's not actually the case. There's heterogeneity. And there's also heterogeneity, we know this, many of you do, in the actual data, which they basically kind of just throw out because it doesn't get to the science paper. And then they're like, next target. So anyway, and there's wonderful people who are working on trying to extract data using AI. Anand also mentioned this, they're working on it in many places, including the Flatiron Institute, to try to gain dynamics out of that data. But here, what we have seen is we can do it also with simulation. And so they say the ability of flu A20 to confer in vivo protection, which people care about because influenza also is coming toward us probably with H5N1, but that's a story for another day. But you know, it is important that we have protective antibodies and so forth. Strongly suggests that HA molecules are dynamic and more heterogeneous in their conformations than we've observed previously, and this is known as breathing. So that's cool, we sample breathing, but for whatever reason, it's probably sampling limited, I don't know, but maybe not, we only seem to sample it in these complex scenes. We have done for years, for a decade, my group had done, and others had done simulations of hemagglutinin and neuraminidase on their own in dilute solution, and you don't see this type of breathing motion. But here we do in this. So this is the flu A20. If you try to dock it into the nice, beautiful science structure, you see it doesn't fit. But here in our open structure, you can see that it actually is able to capably bind. Okay. Now I'm going to talk a little bit about our efforts at the next scale, which I think we can call macro scale. And I don't highlight the whole word because we're still doing analysis here, but it also relates to COVID. So, and just in general, respiratory aerosols. So there's a whole other part of my group that actually works on aerosols, some coming from the ocean because it's an amazing source of what they call sea spray aerosols. But also we make aerosols, respiratory aerosols, when we speak or sing we emit them. And so, we also make these things called droplets. Droplets are large particles that have enough mass that they basically fall to the ground within 6 feet. And this is where this whole 6 feet guidance came from with the distancing. However, there's a large fraction of aerosols or droplets that are small. And these guys they basically they can float in the air for a really long time. And they are, unfortunately for us, if a person is infected with COVID, it's these small aerosols that are enriched with the virus. So it poses a major issue for disease transmission and was one of the big reasons why we just fell on our faces with regard to trying to contain transmission. But now, okay, I got to tell you this. We have these amazing aerosol scientists. These are people who are just spectacular. Physical chemists, environmental chemists. They have all sorts of tools to understand like what these aerosols, what's inside them. Very hard to actually visualize an aerosol, to actually get a structure of it. Really is at the edge of capabilities even now. So there's a science paper in 2020, over 1,000 citations, literally written by aerosol scientists, like the best in the business. And this is the picture they put into the article. And I will say, so one interesting thing, this was an improvement because a lot of people actually thought that the virus would just spray out like naked virus in the air. And so this is like telling people it's actually in something, but it makes it look like it's in just, I don't know, maybe these are ions, but that they're sort of very separated from each other. But we know actually from actually studies in the ocean. So there are amazing parallels between the ocean surface and the surface of our lungs, it turns out. The same type of aerosol production mechanisms are at play. And so what we learned about the ocean, we can see happening in the lung, you have lung surfactant. Your lung is an air-water interface, right? And it has lung surfactant. There are these amazing molecules called mucins, which I wish I could just give you a whole talk about, but I can't. And other things in here, surfactants, proteins, free-floating lipids. And when your lungs expand, it forms these sort of filaments, and then these guys basically break off. And when they break off, oops, I did it again. It's not just the virus that comes off, but it's like a little package of stuff also, including mucins and surfactants, et cetera. And so this is actually a much more realistic picture of what a virus-laden aerosol looks like. And even here, we're hiding some of the things because it's so packed. And so one of my favorite things about simulation and this technique is where I think there's so much potential, also potentially for AI, like we have the ability to, so humans, like we showed this picture of the aerosol, it's so hard for us to understand, even two things coming together, okay, three things coming together, all right. Now we're talking about at least 1 billion different molecules. They're moving. It's super heterogeneous. And how, like, human intuition basically breaks down. So this is, I think, a place where simulation has a lot to give. But, it's hard work because it's 1 billion atoms. So it's basically a nightmare to work with. So what's in a respiratory aerosol? I already kind of walked you through this. You have the virus if someone's infected, and the virus has all these different components like I showed you before. Also the membrane, I could give a whole talk just on how we build that damn membrane. Very, 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 very hard. One of the biggest challenges for cells like simulations is getting the membrane right with all atoms. It's just still very, very hard. You have these mucins. We had to develop these sort of as a first in class to actually build this thing, albumin, et cetera, et cetera. And then there's all this amazing physical chemistry that happens when you bring this stuff together. What we're interested in, one of the questions, we know that the environment is destructive, okay? But somehow the virus will remain able to infect. And like, why is that? Okay, so, a person emits these aerosols, and then these aerosols equilibrate with the atmosphere, and still somehow people are inhaling them and they can get infected. But here's the aerosol, and let me tell you, it gets attacked by light, it gets attacked by atmospheric gases, including ozone, it dehydrates, all sorts of crazy stuff happens. So like, how is it possible that that any of these viruses, also measles, the viruses have like a range of airborne ability. What controls that? Those are some of the questions that I think we can start to, ask and answer now. You already saw this. This is, the simulation, the wiggling and jiggling. You can see it. Yeah, you can see it in there with all the different guys. Okay. I'll say that this simulation was one of the ones one of the sort of the driving use cases for building the new machine that's going to be attacked. So this is like the next exascale machine. The so-called leadership class computing facility. We've used up Summit as much as we could. So these simulations, when we run them, they basically have to take over the whole computer, or at least half. And then when you're using a program like NAMD2 or maybe NAMD3, depending on exactly where you're running it, you can get fairly good scaling, but it's still very, very compute hungry. Anyway, so then you get these enormous data sets. And then it's just a playground of fun things to, analyze and look at. So you can do things like here's aerosol. We took one example is taking a slice through it, and then just looking at the diffusion coefficient of water in here and seeing like where it's moving. So you can see it, it is heterogeneous. When waters get around mucins, spike proteins, or albumins, they basically slow down diffusion. Okay, that makes sense. Our control for water is the inside of the virus, because guess what? We didn't pack it with anything. It's just filled with like water and ions. That's like a project for another day. But we were mostly interested in questions on the exterior. What we see, similar to what happened with influenza, we see that with the hemagglutinin, we actually see that the interactions and dynamics of the spike protein change in the crowded aerosol. So, the spike protein, one of the unique things about it, so it has that ectodomain, but it also has this long bit called a stalk. And Gerhard Humer and Martin Beck made these beautiful cryo-ET and simulation work back in 2020. They sort of renamed these hinges, the hip, the knee, and the ankle, and they showed that actually that when the virus is packed into its when it's sort of packed together tightly with other molecules, including on its surface, like other spike proteins, you see appreciable bending of these joints. And so here, what I'm showing down here, this is the hip angle distribution for the single simulations that we did, because pretty much for all these proteins, as I mentioned, we run like sort of vanilla dynamics of each protein in a dilute solution. It shifts the hip in what we call the RAV, the respiratory aerosol. It shifts over, and it's the same with the knee. So all these joints basically are changing. We're still analyzing what's happening up here in the head domains, to be honest, so I didn't put that in there. This is all unpublished stuff. Okay, and then here's another view of the virus packed with mucins. I mean, it's just incredible to me. These mucins are very negative. They do have negative charge, okay? So you're looking at that. One of the things that we had discovered, actually, was that over time, the virus has been becoming more electrostatically positive. So if you look at the original, let's call the wild type that was circulating in 2020, and you just did the formal charge at pH 7, what you saw was that it had a negative 11 overall charge. When Delta came, it was +4. When Omicron came, it's +10, and it's gotten more and more so. And it's not just that it's actually remapping, if you will, the electrostatic surface. And we showed in other work, which I didn't cite here, that actually that helps, it makes it drive faster to the host cell glycocalyx. But what we also think is the possibility, and these are the hypotheses that we're trying to test now in collaboration with aerosol chemists, is that the virus has basically also potentially been creating stronger interactions with the negatively charged mucins that might also make it more resilient in the air. Because we know from decades of structural biology, if you put a protein at the air-water interface, that thing gets completely destroyed and distorted. So something is protecting that, at least some, not maybe all of them. Some of these viruses probably go to the surface and get destroyed, but on the inside, there's something probably protecting it. We think it's mucins. Last thing I'll say, we're at 977 in here. So ambient CO2. So the other things that we can do with these simulations is understand how ambient CO2 or carbon dioxide affects infectivity. So if you know me, I'm obnoxious. I travel around with a carbon dioxide detector, and it's a measure basically of how much air you're rebreathing. This room is okay. It's not bad. It's at 998, but I'm up here at the front. If we took a measurement in the middle of the audience, might be a little higher. If it goes over 1,000, you might want to start masking. Is generally how I think about it. But listen to this little last bit, and then I let you go. We can ask questions. One of the interesting things that happens is when the virus is inside your lungs are a high carbon dioxide environment, so it has a lot of dissolved carbon dioxide inside the aerosols that come out. So when you emit these, now you have a low carbon dioxide environment, most cases, for example, if you're outside or in a well-ventilated place inside. And then what happens is that you have off-gassing of the carbon dioxide, and that drives pH up. It drives it up to like pH 12 or 13. This is really cool work if you're interested in this from a fantastic investigator at Bristol. His name is Jonathan Reed, his group. And then if you imagine what happens to this virus, if it instantly goes or very quickly, rapidly goes to a pH of 12, there's all these PK titratable residues. Every red spot that you see here is a residue that takes on a negative charge at those different points. Okay, and then so basically what happens is that, and I think, yeah, this is going to play. If you do introduce all of these negative charges, the first structural deformation that you start to see is the membrane just gets wrecked. And so you get these small holes forming almost immediately in the membrane. So we think what's happening is that high carbon dioxide in these poorly ventilated rooms actually makes the virus more resilient to structural damage, because if it's high CO2, there's less off-gassing, and then the pH stays more like it was in a more sort of towards neutral. So this is just a reminder to open the windows. Okay, with that, so seeker acknowledgements, because I talked to you about a lot of things. There's a whole team, seeker, Anand, you heard about these folks are all amazing. I don't have time to say their names. The virus stuff also was like a worldwide collaboration, at least in the beginning. This was the work for the immunogen design. Fantastic collaboration sponsored by the Bill and Melinda Gates Foundation. And then the aerosol work also was just like a monumental effort. It was part of the 2021 Gordon Bell finalist stuff. And, you know, it's like team science all the way. So we're hoping those science papers. So the Gordon Bell paper came out like 4 years ago in October. But the science papers, I think, are just going to come out soon. So thank you for your attention. I'm happy to chat and discuss. Ooh. Do we have time for a minute?
That was fantastic. Absolutely. We have time for questions. Yeah.
Yeah. Yeah, so the question was about the glycans in the closed conformation of the spike, and really we think they're just masking. They're just masking, and the spike actually has several different closed conformations. The conformation that they originally got from symmetry in the original experiments in 2020 from the Wiesler group, you can actually see a little bit of exposure. But then later they actually found these really sort of tightly closed where it's completely shielded. So that just like completely abrogates the ability of the protein to interact with ACE2. Other questions?
One back here.
We really do not. It's something about the transfer. They call that, you know, transfer from the bulk to the aerosol phase. I've been trying to get my collaborators to do the experiments under different conditions. It could also, it's very likely that it varies on a person-to-person basis because of the differences that we have, you know, in our bodies. But that's something that, yeah, we don't exactly know why.
We'll do one more question.
Yeah. Yeah, so I mean, I think, so the question is, why are some people more infectious? I mean, part of it is definitely, and with the super spreaders also, they seem to now think that it's that some people just emit a heck of a lot of aerosols, or they just emit a lot of the virus-laden aerosols, and other people don't. So this is getting back to the question about transferability and how that varies person to person. And it's just, you know, maybe you're in a household and you're very lucky, or maybe you are not at all. So anyway.
Well, let's thank Rommie again.